Hello, today we're going to talk about glucose catabolism. As we know, catabolism is a form of metabolism known for breaking down molecules and releasing energy. Here we will see how energies break down glucose and generate ATP and energy. This process is essential for empowering everything cells do. And we're going to go ahead and take a look at the key stages. Stages that include glycolysis, the Krebs cycle, and the electron transfer chain, as well as fermentation if the cell is either an anaerobic cell or if it's scarce in oxygen. It's an awesome step that they can take in order to survive depending on their environment. So we're going to go ahead and get started with glycolysis. Glycolysis is an anaerobic process, so it doesn't need oxygen to work, and it's a fast way to get little energy from glucose, and it also prepares the cell for the next stages. In glycolysis, we can see that we have an input of glucose, an input of NAD plus and NADP. So here we have our glucose molecule consisting of 6 carbon molecules. It will be broken in half to create 2 pyruvates, each consisting of 3 carbon molecules. We can see that in that process, ADP turned into ATP, our main goal here, and NAD plus turned into NADH. And NADH is an electron carrier, which is also extremely beneficial in glucose catabolism. So depending on the cell, it will either go through the transition step or it will go straight into fermentation. Fermentation is an anaerobic process, which means it happens when there is no oxygen, just like glycolysis. It is an essential backup for cells to generate energy in anaerobic conditions or if they are anaerobic cells themselves. Even though it is less efficient than aerobic respiration, it allows cells to survive when oxygen is scarce. Fermentation for cells who require oxygen is an alternative pathway, and there are 2 main types of fermentation. There's lactic acid fermentation or alcohol fermentation, and it just depends on the cell on which one it generates. We'll see here that we had the input of the 2 pyruvate molecules that were generated in glycolysis, and we have an output. We also have an input, of NADH and ATP and we use that energy. NADH is then regenerated to NAD plus, and it will allow the cycle to continue. This is extremely beneficial to cells just because we don't develop that much ATP in glycolysis to begin with. So by regenerating, we can start the process over. If the process can move on, then it will skip fermentation and it will go straight into the transition step. In the transition step, we can see that we input the 2 pyruvates that we developed from glycolysis, and we can see that here. So this is a pathway to cellular respiration. Here we have our 2 pyruvates, our input, and they are broken down into 2 acetyl-CoA molecules and 2 carbon molecules. So carbon dioxide, since it is a gas, it will float away. And then we see here that we did have also an input of NAD+. So that NAD+ is turned into NADH, which is an electron carrier. And then we can see that here too. So the input and the output. We then take our inputs, acetyl-CoA, and move on to the Krebs cycle. So the Krebs cycle is the next step. So it's the third step here. And we have our acetyl-CoA that we input. And this cycle produces a little bit more energy and loads up on electron carriers, which is crucial for the next step, as we'll see. So acetyl-CoA will break apart and CO2 will fly away. So we'll see that. That is our input. Our output is NADH, which we can see here. The CO2, which is also an output, will float away because that is the gas. We also see that we've developed FADH2, and that is also an electron carrier, and it's similar to NADH. So here we can see that we've stocked up on electron carriers and more ATP, which will be beneficial in our last step. So now, this is where the real energy come from, or the most abundant, we can say. We can move on to the electron transport chain, and we'll take what we developed or we received from the Krebs cycle, which is NADH and FADH. So we can go ahead and move those all the way. Great. And we will actually also use what we developed in the transition step here, as well as glycolysis. So we can see how all of these steps work together in order to combine into the next step, which is the electron transfer chain, also known as oxidative phosphorylation. So here we see that all of these electron carriers carry all of these electrons into the last step, which would be step number 4. And then here we can see what the electron transfer chain consists of. We have 3 electron transport proteins and ATP synthase, because it is an enzyme. So here, let's see here. In the electron transport chain, NADH and FADH donate electrons, which we see here, and they travel through a series of proteins. So the electron transfer proteins, as they move in protons that we see here, so H plus is a proton, are pumped across the membrane. So they are pumped all the way up as the electrons are pumped as well. They get excited, they generate this electricity and push the hydrogen or protons, also known as protons, up, and they create a gradient because they produce an abundance of proton on one side of the membrane. So H plus ions are pumped across the membrane, and like we said, they create the gradient, and it's kind of like charging a battery here. So you have a lot of power source up here, a lot of protons. This gradient drives the ATP synthase, as we said, synthase, because it is an enzyme. Beautiful. And the ATP synthase is a protein that makes ATP, which is amazing. For every glucose molecule, we can see and produce around 28 to 32 ATPs. So depending on the cell, we will either get oxygen. If it is an aerobic respiration, we will get oxygen to combine, and as we can see here, we have H2O. So that's water. That is the final step, and we have this abundance of ATP that will go out to use its energy to perform all kinds of cell work. However, it can also be that the last electron acceptor is not oxygen, and it is the anaerobic acceptor. So any acceptor that is not oxygen. Yeah, so that is glucose catabolism. It is an amazing process that all cells do, and it just depends on the cell and where they go to. It was really amazing to learn, and I hope the process, although it's lengthy, it is good to review. Okay, thanks, bye.